Type I interferon is produced by immune cells in response to viral infection, which acts on various cells to confer resistance to virus. Interestingly, even under steady-state condition, type I interferon is persistently produced in trace amounts, which is so-called physiologic type I interferon. We have found that this physiologic type I interferon is a stress on hematopoietic stem cells and intestinal stem cells.
Binding of type I interferon to its cell surface receptor triggers the formation of ISGS3, a transcription factor complex that transmits type I interferon signals. ISGS3 translocates into the nucleus and binds to the genomic elements called ISRE present in the promoter of interferon-stimulated genes, ISGs. IRF2 is a transcription factor that is constitutively present in the nucleus at the protein level, but is further induced by type I interferon stimulation and binds to ISRE compatibly with ISGS3 to suppress the overexpression of ISGs. Therefore, in IRF2 deficient mice, this negative feedback system does not work and ISG expression becomes excessive. Quiescence is essential for hematopoietic stem cells, HSCs, to maintain their number and function. However, in IRF2 deficient mice, physiologic type I interferon triggers cell cycle entry and the loss of stem cell potential in HSCs. Eventually, the HSC pool is lost. Based on this finding, we wondered, does the same mechanism exist in other tissue stem cells? Thus, we investigated the intestinal stem cells ISCs. At steady state, IR2 deficient mice had a normal epithelial structure in the small intestine, suggesting that IR2 is dispensable for the basal homeostasis of gut epithelium. However, in IR2 deficient mice had significantly fewer ISCs than normal mice. To further test regeneration capacity of the intestinal epithelium, control mice and IR2 deficient mice were injected with 5-fluorouracil to induce intestinal epithelial damage. Although the epithelial structure of intestine in control mice was completely restored within 6 days, it remained impaired in IR2 deficient mice. Thus, the physiologic type I interferon is a stress and impairs regeneration capacity of ISCs in IR2 deficient mice. In normal mice, ISC maintain the balance between self-renewal and differentiation. In contrast, IR2 deficient mice, immature Paneth cells were highly accumulated at the crypt base, suggesting that IR2 protects ISCs from physiologic type I interferon-induced aberrant differentiation into the secretory-cell lineage that causes ISC reduction.
Physiologic type I interferon moderately stimulates immune cells so that the immune response can be triggered immediately after a viral infection. Although this ready-to-go condition has positive effects on the immune system, it has negative effects on the stem cell maintenance.